So thank you very much for the opportunity to be here this afternoon. Just as a means of introduction, my name is Roberto Calle. I'm with Pfizer Internal Medicine Research Unit based in Cambridge. I'm a clinical lead in early development, and I'd like my colleague to introduce himself.
Yes, thank you for having me. My name is Nadir. I'm with Sanofi in clinical operations, and I am responsible for some of the clinical digital innovation portfolio that we are conducting. And I've been part of this VERKKO trial that we've done a couple of years ago.
So we'll get started now by discussing the case study. But once we conclude that, we're going to have a little split. I believe we have a pause for a break, and then we'll proceed with the panel discussion. So what we are going to talk about is remote continuous glycemic monitoring in the context of diabetes drug development, not as a tool for clinical management. And, one might wonder, why do we need to be thinking about innovative ways to look at early development in diabetes trials? Because we have so many drugs for the treatment of type 2 diabetes. And it bears reminding that it still is a huge problem. So we have about 30 million people in the United States with diabetes. Of those, 7 million doesn't even know. They don't even know that they have it. It is probably a leading cause of the primary cause of death in the US, that is coronary artery disease. 36% of those have some form of chronic kidney disease. About 50% develop neuropathy. About 100% of those with type 1 and about 60% of those with type 2 are going to develop nephropathy. So what we're talking is about a huge epidemiologic problem that, in spite of many drugs, only about 50% of the patients actually reach the targets that are recommended by professional societies. So the reasons are complex. It's not just not having the right drug. There are many other reasons why we are not where we want to be. But one of them is we don't really have yet the best drug we could have for type 2 diabetes, and certainly for type 1 diabetes. We're still using new versions of something that was discovered about 100 years ago, namely hormonal replacement with insulin, and trying to the best of our abilities to come up with an artificial pancreas that can actually monitor, like we're going to talk about, and in a non-contiguous way, without depending on external checkups, feedback into a system that pumps insulin into the body of these type 1 patients. We don't have any other means to treat these patients except with insulin. So we're very far from having solved diabetes, either for type 2 or for type 1. So let's talk a little bit about what is the proposition here, what is the case study. And what we want to talk about is the use of continuous glucose monitoring in the context of early drug development for dose selection. So what is our main target in early development, and particularly for new drugs for diabetes? Well, it is to fully characterize the pharmacokinetics and the pharmacodynamic response profile of this new drug for these patients. We have very clear guidance for the approval of new antidiabetic agents. The measurement par excellence is hemoglobin A1C. We also make use during the development process of direct measurements of glucose, first by direct measurements with an analyzer. Later on, we use self-monitoring of blood glucose through finger sticks from capillary blood glucose. So what is missing in these measurements for early development? Well, the great advantage of hemoglobin A1C late in development is that it gives us an integrated measure, right, of glycemic control. So it gives us an idea of what's happening over time without having to test those patients repeatedly and frequently. But that same advantage is a disadvantage when you're looking at it early on in development and you're trying to decide what is the best drug for, or what's the best dose for a particular drug. You don't want to be treating somebody for weeks before you want to decide, okay, do I need to adjust the dose or not? You need to make decisions promptly. So that's where monitoring glucose is used. In phase 1, analyte measured glucose is commonly used in the phase 1 studies where part of the time at least those patients are inpatient, so you can monitor fairly regularly by venipuncture. As you're moving a little further along, the patients are going outside of the clinical research unit. You're still monitoring them, so you're going to be using more the self-monitoring blood glucose through capillary sampling and monitors that are used as an outpatient. The limitation then again is richness of the sampling. So you can only ask somebody to prick their fingers so many times in a day before they say, okay, enough. And even in the unit, they're only going to let you do venipunctures only so many times until they say, okay, that's enough. And the more information we have, the better decisions that we can make. So what we want to do is enable a richer sampling that can better inform our decision making. So if we're able to do that, we can improve the efficiency of drug development by increasing the volume of data collected. With more data points, we get a more comprehensive profile, a better time course of what's happening with each individual patient, and we even can monitor at times where otherwise we could not monitor, during sleep, during exercise, times when you're not going to be able to be doing self-monitoring or doing a venipuncture. So it is a much richer data set on which to base decisions. In addition, almost by default, even if that is not your primary goal, it has a built-in effect on safety, and you could potentially enhance safety because you can identify when that glucose level is either excessively elevated or excessively low, and do it very quickly and essentially with real-time or close to real-time information. So what are we talking about now if we put it in context of this use as a biomarker? And what we're talking is, looking, of course, back to the best resource data book, is a pharmacodynamic biomarker so that we can very quickly measure the response of this new anti-diabetic agent when it's introduced into this patient's regimen. And by default, we immediately also get a safety biomarker that is going to tell us when we are moving outside of bounds. But we're going to focus largely on the pharmacodynamics biomarker as our goal. The safety, of course, we will mention because it's unavoidable, but it's not the main piece of the conversation for today. So, context of use. To come up with the right context of use, we need to be able to inform, so what is the decision that is going to be made? Who's going to be informed? What is the situation, the clinical situation on which that particular decision is going to be made, right? So I actually like this next slide because we look at the actual anatomy of a context of use. First, you define what's the category of the biomarker. Then, what's the purpose? In this case, drug development, under which circumstances? That is, in early drug development. For what type of disease? Diabetes. And what type of decision? Dose selection. So that context of use then would read something like a pharmacodynamic biomarker used for the purpose of dose selection, that is, determine optimal dose for a particular drug, in a clinical study with participants with diabetes receiving an experimental anti-diabetic agent in an early drug development phase 1, phase 2 trial. That's what that COU would look like. Now, This case study that we have put together for you was inspired by this study, the VERKKO trial, that was run by Sanofi, Mendor, and eClinical Health. It's not a continuous glucose monitoring study, but it is a remote monitoring study for glucose. So for more details, I leave you with Dr. Ammour, who actually ran that study.
Thank you, Roberto. So this is a study that we've conducted in 2015. And the questions that we had at that time was, can we engage patients remotely, reducing the burden that they have to, when they go into the clinical study, to come to the clinical site every 3 weeks, every 4 weeks, for the measurements that is regularly done in clinical trials in diabetes. So we had, at that time, the three other companies, Mendor and Clinical Health. Mendor is a glucometers manufacturers. They developed a glucometers that is very simple to use. It has a 3G chip into it, and when it gets connected to the wireless bands, it can send the signals directly into a platform that was provided by eClinical Health. So the question was, if we are able to engage patients remotely, get them into a trial without having them to come to the clinic, can we reach out patients that will usually not get into clinical trials? Can we get patients into clinical trials that will be much more convenient for them so that we can collect more data more easily? And can we do that in a way that is totally acceptable from an ethical and regulatory perspective? So that study was designed to achieve two objectives. The first one was really to validate that wireless glucometers in its ability to collect correct measurements and send those measurements through the platforms to be reviewed by a physician who is on the other side, can monitor the progress of the patients and their glucose levels. But secondarily was also to evaluate the feasibility of recruiting patients online and get them through the procedures and the training procedures totally independently from having someone sitting next to them and showing them how to proceed and how to proceed with all the procedures that are needed to do that. That study was conducted with a physical team from the Finnish Helsinki Diabetes Center, and the principal investigator was associated with the design of the study. And that involved one study coordinator, a nurse actually, into the study. And we were looking at recruiting patients around Helsinki in a radius of 50 kilometers around the city. The study started in November 2013, which is which is the date where we received the ethical committee approvals. There wasn't any specific questions. The protocol was designed in a way that in case of specific conditions, the investigators can contact the patients and get them to the clinic to a physical examination. If there is no specific signals, they can just continue the study remotely without having to come to the clinic. And no patients in that study had to come to the clinic for the physical examinations or clinical examinations. We recruited 50 patients, which was the target, and it took 5 months to get those 50 patients. The recruitment was done through a campaign in Facebook and using the digital agency that was conducting the screening facing patients facing screenings and so on. Now, when we looked at the first component was how many patients would actually manifest the interest to get into the study. And we had 74 patients that were manifested the interest to get into the study. They get a screening page. Are you interested to participate into an experimentation study? 74 patients have responded yes. And amongst those 74 patients, 50 have been meeting all the criteria that were very simple. And most of them were required to have their own internet connections at home and nothing else. There was no smartphone involved. It was really the ability to follow some procedures online and conduct that online. They get through the consenting process online. The platform has those features. the investigators was reviewing the questions from the patients. There was a couple of quizzes to make sure the patient understand what they are consenting to. And after the consenting process, the platforms was able to ship the glucometer to the patient address. And when the patient received the glucometers at home, they need to log on into the platform again and follow a series of procedures to activate the glucometers and go through some basic trainings on how to use it. It was the new glucometers. It wasn't glucometers that were used to use, on their daily practice. And after that, they had a procedure to complete a full, I think it was a 7-point glucose profiling study, 2 times within 2 weeks. And it was a replication of a study that was done using traditional methods. Just for your information, in the traditional study, it took for 1/3 of the patients, it was around the same size of patients. For 1/3 of those patients, it took 3 attempts to get the right glucose profiling. Why is this? Because they do the profiling at home. They record all of that into the paper diary. The next week, they come to the clinic. The physician will check whether they've done that correctly. Some of them have not done that correctly. They get retrained. They go back another week. They do it again. And again, few of them were still, after 2 weeks, not capable of doing it properly. So they had a 3rd week to do it so that they can get the full measurements for all the patients. So in that study was replicating that. And 51 patients have completed they've started their glucose profiling. There was 2 patients that were unable to perform that correctly. The physicians and the nurse was able to monitor that on a daily basis, and they were able to identify those patients immediately, call them, and get them just clarifications point over the phone, and the day after the patient was performing that correctly. So it was very, very clear and an appropriate way to identify the patient who may need additional assistance, additional clarifications, without losing another week or 2 weeks to do that. Interestingly, we had at that time a lot of questions about, is this kind of study appropriate for the type of patients? And there is a kind of counterintuitions that technology works for younger generations, but when you get into the older populations, they may be not at ease with that. Actually, the average year for that was 59, as you can see on the slides. And most of those patients, actually, there was few of them that were under the 50, but most of them were above 50. And the reason is because they have time, and they have, the interest in their healthcare is much higher than the people who are younger. And they were very interested in that concept to be participating remotely. I will share also some other news on that, actually. So a few observations on the positive notes. The IRB has been approved without any specific questions. I think the protocol was simple enough, and that's an important point when we design this kind of study. You should not go into very complex procedures. It has to be very simple, very straightforward, designed for patients that are not expert, they're not medical experts, they are patients. So those are the things that needs to be considered with that. There was a couple of feasibility studies that was done so that all of that was tested with real patients before we get into the study itself. The site staff was telling us with these kind of settings, it took them probably 1/3 of the time that they have expected to spend if they had to do it into the normal way. And it was for them the first time they were able to follow 50 patients within, those several months, as opposed to usually in this kind of study, they will do 10, 12 patients maximum because of the workload and the attention that is needed actually to that. From a compliance perspective, as I have said, the few patients who did not perform correctly the glucose monitoring and profiling was catch up very quickly and were corrected very quickly. You don't get bad habits installed in the patient's procedures after 2 to 3 weeks of practice to find out that the patient has not done that properly and have to retrain them. So they get corrected almost immediately. One patient was able to travel while still into the study, and this is one of the comments that he provided back at the end of the study, and he found that very convenient actually to be able to not disturb his normal life, his social life, and still be able to be into the study. And most of the patients have, responded to a survey at the end of the study. A couple of things that come out. They were very satisfied with the fact that technology works without bug, and this is really important that you test that very, very carefully, and you don't expect to have something that works with technical issues. And they were very pleased with the simplicity of the procedures there. However, in terms of extensions to the continuous, glucose monitoring, when we have, glucometers, as we have mentioned earlier, you have limited number of measurements. So you don't have the full measurements that you can get with continuous glucose monitoring that offers superior benefits in that respect. It does, when you have the procedure with the glucometers, even the 7-point profiling, you cannot repeat that every day. You can do that 1 day per week, 2 days per week. That's the maximum you can expect from patients. You cannot expect them to be able to continue that 5 days in a row or even 2 weeks in a row. That's absolutely impossible. You have to rely on the investigators for the patient verifications. This is something that the investigator has done to check that the patient is a real patient on the other side of the platform. So there is some area that still requires some additional clarifications points. I can tell you from experience in our auditing reports that it is not uncommon that patients might sign up into 2 different studies at the same time just to get the drugs and get some other benefits on that. So this is something that really that needs careful attentions. The device should be functioning with no technical issues. This is really important. And in that case, it was a new device and it was trained properly for that. We do need the infrastructures to be working correctly as well. And in particular, when we look at what is the appropriate guidance so that we can deploy these kind of studies more routinely in the practice. But also in that study, it was 15 days. So we haven't looked at whether the patients over a longer period of time will decline into their adherence to the process. But I do believe from talking with patients who have CGMs, if you go into this kind of passive monitoring and passive data collections, it's also an advantage to the patients and help them with their compliance over the course of the study. I will turn back to you, Roberto, for the rest. Thank you.
Thank you, Nadir. So you can see that very interesting trial and actually establishing a proof of principle for running a virtual trial. And although it was not a CGM trial, it did implement this remote monitoring concept, which is something that we were interested in exploring in this exercise for this case study. So with that, let's come back to the case study at hand, namely utilization of CGM and metrics within CGM to have a pharmacodynamic biomarker for the purposes of dose selection in a clinical study with participants that have diabetes receiving an experimental anti-diabetic agent. And the profile that you get is something like this. This is just a cartoon, and I'll show you some more information in a minute. But basically, the profile that you get allows you to see very clearly when the patient is in what is considered the adequate range or the desired range, which in this case is referred to as the time in range. And in fact, what we're proposing is that the biomarker would be precisely that, the time in range for the particular glucose values. The most commonly used range for that is 70 to 180 milligrams per deciliter, but that can be varied. You can change that. But that is what is most commonly used. And then above and below that, you have time above range, time below range, and then categorizations for the severity of the deviation from the time in range as illustrated in the table that you see at the bottom. And you can see graphically very easily when a patient's glucose values are below that desired range or above that desired range. And that brings a very visual, easy to see information about when they are in the safe zone and when they are outside the desired range. This is something that is very much doable right now. We have the technology. There are FDA cleared devices that can do that, and in fact they are used for clinical for clinical management of patients, which is different from what we're trying to do, right? We're not proposing its use for clinical management. We're proposing its use for internal decision-making in an early clinical trial. It's a different use than what these particular devices are being used for. You see there are a number of them. The majority are attached to the skin and have a transducer inserted into the interstitial space in the skin. One of them is actually inserted and implanted in the subcutaneous space. And the purpose of showing you this graph is not to start a comparison between each of these different devices, but just to show you that the technology exists and that it's being used for a particular context, and we should be able, and we're proposing that we use it in a different context. So some of the hurdles that we have to be mindful when we are utilizing continuous glucose monitoring. Well, first of all, what is our reference, right? And our reference usually is going to be either glucose measured from venous blood in an analyzer, or more commonly, if you are in an outpatient setting and self-monitoring, is self-monitoring blood glucose with a capillary sample obtained by a pinprick. But what you do get when you're doing continuous glucose monitoring is actually a measurement of glucose in the interstitial space. Same molecule, it's not the same information, however. So there is a lag time of approximately 10 minutes between the blood and interstitial fluid glucose readings, so you have to be mindful of that when you interpret the data. And there is about 10 to 20% difference in the values that you can get between your reference measurement and what you obtain from your CGM measure. And there are a number of different sources of variability beyond just where you are obtaining the sample. So the insertion site matters and can give you different information, whether you do it in the arm, whether you do it in the abdomen, whether you do it in the thigh, that matters. Also the glucose range, where that patient is functioning. So the best performance is in the middle, but you're going to start to have some deviations, particularly at the lower end and at the upper end, and the characteristics for that performance will vary according to the technology. And then, so all of those need to be integrated into your evaluation when you're trying to reach conclusions based on those data. What you see on your left-hand side is just a listing of the more common metrics. I have highlighted there with the broken line the time in range, probably a bit hard to read from where you're standing, but that refers to that 70 to 180. And then above and below it are the ones that I pointed in that other slide with the categorical groupings for deviations above and below. And what you see to the right side of that is a typical report. And what you get is at the top, you see that bar graph, it's that stacked bar graph, gives you a breakdown of the percentage of time spent in each of those time glucose ranges. So you can get an idea of what that profile of that for that particular patient looks like. And then at the bottom there, you see an integrated measurement back in of what are the average values for that patient and the variability around that if you were to integrate that into a single 24-hour period. So now there's some key things to keep in mind when we're thinking about glycemic control, thinking about the data we get from CGM, how it compares to a mean value of glucose that we obtain in repeated measures, and what we get with a hemoglobin A1C. And one of the things that is striking is that you can have exactly the same mean glucose, and you can have exactly the same hemoglobin A1C value as an integrated measure, but they can represent extremely different profiles for that particular patient. So that's the example right there for you. In the graph, that you see what looks like a sine wave there, that the blue one represents the patient that is represented by the bar graph on the extreme left-hand side, and that patient is spending most of the time above in the above range space. If you look at the light blue, that represents the patient in the middle. So that patient is spending, I don't know, about 2/3 of the time in range, and then spending about almost a third in the above range, and a small portion of time below. And then you have the one in yellow that is essentially spending all of the time in range. All have the same hemoglobin A1C, all have the same mean glucose. So that is the value of being able to monitor with a rich data set as provided by CGM. So as we think about it, and what is the trust that we put into those measures, we need to think about how well it represents the biology of that disease, the pathophysiology of that disease, and how well it actually represents the type of information that we normally would obtain from an integrated measure such as hemoglobin A1C or glucose. So as you all probably know, the way we diagnose diabetes is all based on epidemiology. So that the criteria that we have for glucose, more recently, in recent years, hemoglobin A1C, are all based on a particular risk of developing microvascular complications and a cutoff that actually predicts that particular risk. That's how we do that. We do not have any prospective studies looking at CGM and, for example, time in range and determine, okay, what % time in range actually correlates long-term with microvascular complications. But we do have an approximation. So this is an exercise that was done utilizing data from the DCCT trial. And the DCCT trial is the one that actually finally established the correlation of tight glucose control with improvement in outcomes. So it compared in type 1 diabetics tight insulin control with standard insulin control. They used self-monitoring blood glucose, which they were instructed to do that very often, and periodically they would obtain the 7-point mean plasma glucose that we have mentioned throughout the day a couple times. So you could obtain a more rich pattern. Still nothing like what you can get with CGM. But you can actually calculate an estimated time in range from that 7-point mean plasma glucose. And that's what you see in those graphs. So what is represented there is that the higher your time in range, again, as estimated from 7-point mean plasma glucose, the lower your probabilities of developing complications based on data from the DCCT trial. And that is the closest that we have to make that link between CGM metrics and actual outcomes data. And this is a representation of what you actually can get when you monitor these patients with continuous glucose monitoring. At the top, you can get a very rich descriptive pattern of where this patient is landing every single day, all the time, 24/7. Whether they are active, whether they are resting, whether they are sleeping, whether they're exercising, all the data is integrated there using time in range as your biomarker. And these data can be made almost instantaneously available to a sponsor to analyze if you establish something like what was used in VERKKO, so that you have remote distribution of those data to a central cloud station. And that then can be fed into the pharmacometric models so that you can improve your predictions for what is the optimum dose for that anti-diabetic agent. And that's the proposition, the value proposition that we believe can be made by using CGM in that early development space. So I showed you some of those devices that granted they're developed for clinical use, so let's be clear, not for this particular use. But there are wearable devices that utilize a transcutaneous electrochemical sensor. One of them is implantable in the subcutaneous space. There are multiple manufacturers. There's proprietary technology and proprietary algorithms to provide the information, the type of information that I showed you. But these are existing measures. We're not measuring a new analyte. It's the same analyte. We're looking at this analyte in a different way, capturing much more data, much more frequently, and integrating that data into our information for decision making for dose selection. And these are all FDA cleared devices. As know that we're running a little late, but so I'll try to catch us up as much as I can. In terms of benefit risk assessment, the benefit here is that by providing a richer data set than what you can obtain with SMPG, you can get that improved characterization of the 24-hour glucose profile, build that into the early development space, and having immediate access to that glucose data to analyze, your pharmacometric models can be updated more frequently and faster. So, so with that, you can actually accelerate drug development. So I'm trying to move a little faster as well. So how about the benefit? Who accrues the benefit? And in theory, here you have to both the individual as well as society. For the individual is the ability to participate in the trial in which more data is being obtained of the burden they're taking on as a participant, and with less active involvement from the subject himself or herself. And the technology can serve double duty as a safety biomarker if we so choose to do by activating alarms, which we don't need to do if we don't want to. We can rely on the more standard monitoring as MDG for that purpose if we choose to do so. For society, is the improved ability to accurately ascertain an optimal dose is a benefit to society. Now we have a drug that by selecting that dose should be safer, more efficient use of that drug, and having a more efficient drug development process should translate in lower costs, and that means that those resources can be deployed where in otherwise where else is needed. So, however, of course, there are risks. The fact that we're measuring glucose means we're capturing adverse events as well. We're capturing the adverse events when that patient has hyperglycemia that goes above the range that we have determined, or hypoglycemia when they go below. And in early development, it's probably the hypoglycemia that is going to be the most relevant, because in the short period of time, those are the ones that can get you in the greatest trouble. So the risk is that if the technology is not functioning as expected, is that you end up with an incorrect dose. If you are ending up with an incorrect dose paradigm, you may be overcorrecting or undercorrecting when you measure the glucose in that particular patient. Also, if you decided to activate alarms and you as a sponsor and the patient start to rely on those alarms to identify hypoglycemic episodes, and they're not functioning as they should, and you're not implementing self-monitoring of blood glucose, now you have actually increased the risk for that patient to have an adverse event, particularly for hypoglycemia. So potential mitigation factors here are that you can maintain SMDG for the purposes of safety and just leverage the greater richness of data to get a better decision in terms of dose selection. And also, if you select doses, you're still going to go into late development. You will still see the performance, right? And you will see how well your new drug is performing in terms of controlling glucose compared to older standards. And you will also see how it's performing in terms of safety and frequency of adverse events. But that's exactly what we're trying to avoid, right? We're trying to avoid getting to a stage where you're having late development and you're making corrections because you didn't get the right data in the beginning. So that's why it's important that we have a very accurate and precise measurement in that early development space. In terms of who is informed here, and the ones informed are the sponsor for the internal decision making. The value proposition here is not for a regulatory approval. For those, we still believe that the more standard measurements are the way to go. It's the whole value proposition is in terms of the purpose of accelerating and optimizing the dose selection. And in terms of our own trust in the validity of the biomarker, because we're measuring an analyte that is well known and it is integral to the pathophysiology of the disease, all of these really fall into place very easily. Universality, everybody with dysglycemia will have dysglycemia. No, that is the very definition of diabetes. It obviously has biological plausibility. Causality is not a question. Proportionality in terms of glucose itself is not a question. That's been determined through the DCCT for type 1, through the UK PDS study for type 2. It's about the new way in which we're measuring with time in range and reassuring ourselves that time in range still fits squarely with those different pieces of the puzzle. And here then it's fair to ask, what additional data, if any, do you need to fill that gap? So I'm going to pause here. I think we are ready for a break, so we'll do that. And after that, we will come back with our panel and have a Q&A. Thank you very much. All right, so in the interest of keeping us on time, let's get started with the panel discussion. So before we proceed, we would like to have each of our guest panelists introduce themselves. I'd like to start with my colleague, Joe Mathers, who is joining us online from Pfizer. Joe, can you please introduce yourself?
Yes, my name is Joe Mather. I lead a group within digital medicine and early clinical development at Pfizer.
Thank you. So, Denyse, can we start with you then in this way? Introduce yourself.
Hi, my name is Denyse Niemerov. I'm with Abbott Diabetes Care. I'm a manager of national accounts, and I work with pharmaceutical companies and institutions doing research.
Andrea?
My name is Andrea Bell-Vlasov, and I work for the Food and Drug Administration in CDRH in the Division of Chemistry and Toxicology Devices, and I primarily work on diabetes devices like CGM and automated insulin delivery systems.
Hi, my name is Jagdeep Podichetty. I'm a scientific director with the Critical Path Institute. I'm part of the quantitative medicine program. One of the things we're working on is in the field of type 1 diabetes prevention trial. We are also working on ramping up our efforts with digital and AI, some of the work Diane mentioned in the morning. And I'm excited to be here.
Nadir Ammour from Sanofi. I'm working in clinical operations and involving clinical digital innovations in some of the programs leveraging patient-generated data and electronic health records.
Thank you. And you already know me, Roberto Calle from Pfizer. So, we have a couple of points for discussion and even some slides to support that. But before we do any of that, actually would like to make sure is that we offer the opportunity for the public audience to take the first stab at it. If you have questions, we're open to that right now. Please join us at the microphone.
So go ahead.
Hi, Shamay.
Hello. I had a question regarding off-label use for CGMs. You know, potentially, I don't know, Roberto, from what you showed earlier, how many of those devices were approved in certain populations or if there were restrictions on age, for instance. And then, for the wearable space, there's a lot of off-label use for wearables. I think it's a little bit more regulated in the CGM space. I was just curious if your approach would be different in the clinical trial space because of this, if you would do different kinds of monitoring or just not even use CGM in those kind of contexts if the population wasn't approved for that device.
So those are FDA cleared devices, but I think we have recognized the hurdles of making that translation from the use for clinical management of patients versus using as a research tool. So, and I think that Andrea and Denyse are particularly well prepared for commenting on that, so I'm going to leave you with the two of them.
Yeah, why don't I start? So in this context, we're using CGM to develop a drug. That the problem with using devices off-label is that the performance is unknown in for example, in certain demographics like pediatrics, for example. You're already dealing with a margin of error that these CGMs provide. You're really rolling the dice when you're including different uses where the performance is just simply unknown. So you're really complicating the quality of your biomarker in that instance, and how well it's going to get you to what you need out of your drug development, your trial, whatever you're running. So I don't know if you have anything to add to that.
Well, I have a little bit of a different perspective. From my experience, a lot of people are using CGM off-label. The people that are doing research into better ways to manage diabetes are looking for a better way to understand what's going on with glycemia. And CGM, quite honestly, is the best way to do that. I understand, like Andrea said, a lot of these devices are not approved in pediatrics, but people are using it in pediatrics because they're making the assumption that if it's approved and it's proven effective in an adult population, it's also going to be, proven effective in a pediatric population. But when you look at the burden on the patient in a clinical trial and getting to kind of a, an end game of, how do we better manage this disease, a CGM is definitely the most the least burdensome way to get there. So that's why I think it is being used. Obviously, there's regulatory concerns around that, and the people doing the trial have to make sure that they have informed consent, and it's being disclosed that it's being used off-label.
I want to make a comment on the clinical, the drug development side of things. Using CGM on the drug development side, there is a part of biomarker, using it as a biomarker, for example, like you mentioned, time and range. But then on the other side, when you're using it for dosage selection, then there's an aspect of developing quantitative solutions in order to connect the two. And there is a burden on that in terms of developing those tools as well to connect the two things.
So acknowledging those gaps. So maybe from the point of view of, and if I can lean on the two of you again, from the point of view of a manufacturer of a device, can you comment on what can you do to enable that? And Andrea, maybe you can tell us a little bit about what would it take from a regulatory point of view to convince regulators that this is actually a feasible and valid way to go about making these decisions? Maybe.
Yeah, I think from a device manufacturer perspective, what we need to do is validate that continuous glucose monitoring data is a good measure of diabetes management success. We know A1C has been the gold standard for years. As Bakul mentioned earlier, it's the not so gold standard anymore. Time and range is now kind of the qualifier to A1C. But we still need to do those studies to really prove that out. Roberto in his presentation showed the correlations from the DCCT trial. Now that, wasn't a perfect measurement, but it was going back a number of years before CGM was really being used and looking at finger stick testing and trying to take that data and fit it into a time and range measurement. And we did see correlation. So I'm confident that as manufacturers, we would see that correlation between improved time and range and reduced negative complications and outcomes, or better outcomes. But we need to put the work in to do that. I recognize that.
Yeah, I agree. I think that we don't know enough about, time, for example, time and range and what the, associated clinical outcomes are. If we're presented with that information, I think things may change, in terms of, for example, right now with CGMs and automated insulin delivery systems, there's not really any efficacy claims. They're approved based on and cleared based on safety, with the understanding that these devices allow people to manage their disease more easily. If we had, more information, then maybe down the line in the future, there could be some claims, but that, as you just, stated, that information is lacking. Or simply, for us, it's just not there yet. Mm-hmm. So, that's what we would need, the community to kind of help define why these biomarkers are useful and what kind of outcomes these lead to, what benefits they're tied to.
Can I make
Go ahead, Joe.
a comment? And what I'm hearing, and maybe I would like you to comment on this, is kind of a connection that I'm not sure the COU derives. And because we're talking about outcomes now and whether time and range actually has an effect on microvasculature effects or whatever. The COU was dose selection, right? Which has nothing to do with outcomes, really. Correct. I mean, it has to do with a decision that's based on whether the company wants to go higher or not, right? Right. So how many, and so now these are the kind of conversations I feel like I have with my teams all the time. And so I'm wondering, how do we, here are a group of experts that are also kind of putting the two together, and I'm trying to understand how can we make the COU or make the discussion stay focused on, what it is that we think we want to do with in the drug development tool. And then, as Bakul had talked about today, I mean, he showed, and I'm going to steal those slides because I do that frequently, the slides where we are reiterating and building more data and generating more and more complex device information. And separate those two and say, okay, this is where we are, and move that. How do we ensure that the conversation stays focused?
Yeah. So actually, the way that COU was built was precisely to try to stay away from those discussions, but it's hard because it immediately comes to mind because we know that we can get so much more information from that type of measure. But to your point, we just try to bring it back to that. Maybe we can use one slide. If you can bring the first slide in the backup. Oh, do I have control? Oh, great, perfect. Thank you. So I figured, for the purposes of discussion and not to suggest that this is a perfect design, I figured we could put together for you a hypothetical dose ranging study design. And how one could envision this working to optimize dose selection. So in this case, we're talking about, I'm not specifying type 1 or type 2, but let's assume a diabetes population, early study, maybe a phase 2A study, where we have done some initial dose ranging, But we are now with the objective of optimizing the guidance that we have to give for that drug in order to have the right dose selection. So, for example, you can start with a cohort of patients that can be, some in placebo, some in active, doses as determined with the assumptions that we bring into our pharmacometric models from the dose escalation models, and we start there. And let's take 8 patients, 10 patients per cohort, per arm, and we treat them for a period of time, 7 days, for example. You're capturing data with the CGM, and you are transmitting it directly, right away, to the sponsor. Period 1, all of that data has been sent to the sponsor. The sponsor has been feeding their pharmacometric modeling and comes up with an improved model for projection of doses, and with that selects new doses or new guidance for selecting the doses based on the glucose of the patient, and implements that in period 2, same patient. So now you have the same patient being its own control, granted. Sequential, there's a time dependence effect, all of that is acknowledged. Second period, you capture more information. Take it directly into your pharmacometrics model because you're remotely taking all of that data in. Again, inform the pharmacometrics models, come up with a new prediction, do it for a third time in period C. And at that period of time, you update your whole model. You have a new guidance of how you're going to adjust the doses according to the patient's glucose, and start a new cohort. Because that was based on a particular group of patients. Now, okay, how well does that work in a different group of patients? And now you start the whole process again. Period A, capture all of the data, update your pharmacometric model. Period B, update the pharmacometric model. Period C, now update the whole model and start a third cohort. And by the end of that third cohort, you have multiple sets of data richly informed by CGM in 3 different groups, 3 different groups of populations, same entry criteria. This is one single study. And at the end, you end up with a guidance for dosing that is based on all of that information. So it's sort of like an adaptive design for an early trial, because you're changing the dose based on emerging information. So that's the concept, something like that. So why don't we keep the discussion on that basis? Joe, is that more or less what you, okay. The discussions about the regulatory pieces are much more interesting, Joe.
Discussion.
No,
it's just,
all right.
but
so
what is remind me.
Yeah.
In the end, this is phase 1. Like,
yeah, phase 2.
Kind of, I mean, for 1, 2. In the end, when you're going into your registrational trial, you'll pick 1 dose, 2 doses. I'm trying to get what decision are you trying to make with the guidance that will end up.
So it depends on the type of drug you're doing. Let's assume for a minute that we're talking, not that we need another one, but let's say that we're using a new short-acting insulin, right? So you have to come up with what is the exact guidance for a particular level of increase in glycemia, how you're going to adjust that dose, right? So that's what you're trying to get at there. What is the right metric for adjusting the dose depending on the response of the patient? So this is really meant more for a short-acting drug that needs adjustment real time. For example, like I said, let's say you have somebody that is being treated with a background basal insulin, and you are coming on with either a new short-acting insulin or some other drug that is going to modify or modulate the acute changes in glucose, for example. So you need to understand how to adjust it to maximize that time in range. So that's what this would give you.
Yeah, I mean, in theory, it's pretty good. I mean, you're creating a feedback loop where you can update your parameters in your pharmacometrics model and then optimize the dose for essentially all three cohorts here, of patients in all three cohorts.
Mm-hmm. Yeah. So this doesn't get into the problems or discussions of its use in late development. But I know that there's a lot of interest in that. There were some questions during the break that were precisely going into that space. So up to you whether you want to open it and allow some of that discussion. But I think there were some interesting questions raised there that might be worth.
I think as long as we're moving from one space to another space with our eyes open.
Yep. All right. So, so I know some of the folks had some questions over the break that they wanted to bring up. So please feel free to chime in.
One can easily imagine to go beyond the specific use case of a short-term correction dose finding to the broader question, which is potentially very exciting here, which I'm sure is in people's minds who have these machines, and that is the broader societal question and health for the population. And this gets into the argument for groups of people sharing data more quickly, I think, to get to Dr. Niemerov's point about if you could really show that a tighter control of the excursions of glucose during the day is possible, not necessarily with insulin, but some other novel treatment that somehow helps modulate the system, which I'm sure is the dream of a lot of drug developers, then this would have huge implications for testing that apparent finding, which looked pretty convincing the way it was presented, that you get these microvascular changes associated with poor control of diurnal glucose, right? And the reason I got particularly excited about this is because other discussions are going on about the importance of metabolic control in other conditions, one of which is of huge interest to a lot of people, and that's Alzheimer's. So just to share this, one of the conversations going on about whether or not one should put together a large prospective effort to clamp, to actually make a societal investment in better glucose control and looking at other long-term outcomes, obviously nephrotic disease and everything else. But is this tool, what I'm really asking is, going pretty far out, is this tool good enough to justify thinking about such a big study? Okay, because I mean, the short-term application that Roberto laid out is beautiful. I mean, I can well see that for internal decision-making in a company. I mean, that would be fantastic. But should we be looking at this as one device out there which could have a much wider use? This may be like asking the choir or something, but it certainly lights up some possibilities.
I'll make a comment here, and this goes back to the original question about off-label use, but I know that researchers across the country are looking at the impact of glucose variability on everything, from Alzheimer's, from cancer, to healing after surgery. So there is a lot of, there's certainly a case for using products like continuous glucose monitors to determine how glucose affects not only your diabetes and the complications that come from that, but other aspects of overall health.
I just want to add too that, you know, CGM, yes, it's a very powerful tool, but it's also very important to really understand the nuances of the performance of CGMs. And these CGM is, yes, we have CGMs right now that are approved for non-adjunctive use, so patients can dose directly off of the values that are provided by these CGMs. And then we have others where you cannot. So that being said, yes, like there are some that are good enough to dose off of, but there's still a margin of error that you need to understand about these devices. And we still consider the performance of these CGMs to be inferior to BGMs, which also have a margin of error. And so it's going to be really important to define the level of performance that's going to suit your need for developing these types of drugs. So, there's a lot of things to consider, and that goes beyond just the nuances within a certain brand of CGM, but across the board for the device, area in general.
Yeah, so if I hear you correctly, then just as we're doing for other measures in the biomarker pre-competitive consortium space, which is what some of us work in, then perhaps a sort of collaborative look at this might be important if one is really going to decide on whether to implement a tool in population studies. So to that end, is there a path to doing that right now, or is this something that because everybody has their own devices, they're competing and everything else, so there's no way to get this done, or there's no current plan to get this done? So concretely, is there a plan to get this done, or is it all open right now?
Me, I don't really know. I'm just speaking from the device perspective, so I can only tell you, what the issues might be with such a device, but it would be up to the community or, the pharmaceutical developer to determine what, level of performance would be good enough for them.
So the reason I'm highlighting this, so at the FNIH, in the sort of, for instance, I'll just take the proteomic space where the FNIH has done a lot in the pre-competitive space to compare different assays across multiple platforms and help the field decide which have the best performance characteristics to put into larger prospective studies where there's a lot of public and other money going into them. So that's what I'm really asking here.
Yeah.
So Roberto, can I jump in here on?
Please, Joe, go ahead.
I think the other use case that you want to consider for the CGM devices specifically are how these provide context for other measures you're doing. So for instance, in an AD trial, if you're trying to understand information about dietary consumption or glucose utilization based on activity patterns of people, this is a perfect device for ground truth around what's actually happening as compared to, say, food diaries or other reporting devices. So it's one way that you could use this device and its performance characteristics in a population to verify another endpoint that you're measuring for a longitudinal study. So just another concept that we've been kind of playing with in the metabolic space as well.
So, so I actually wanted to take the opportunity since you chimed in. One of the key hurdles that we need to be able to solve in order to implement, even for the early development implementation that we had been discussing, is bringing that data in. So we were talking about not just collecting data that had been stored locally at the end of a study, but to be able to remotely transfer that essentially in real time or very often. And I know you've dealt with that in some of our studies. You know, if you want to comment a bit on the hurdles to manage data, to receive data, to manage missing data, and that sort of thing when you're receiving such a massive amount of information.
So one point I wanted, or question, I guess, thought is, what if someone thought, or that there was data out there that suggested that Alzheimer's progression or diabetes or whatever was associated, could be associated in some way with respiratory rate, all right? Total made up, right? From what Elena talked about today, she had 2 different monitors. Neither one of them compared to, well, with each other, or to someone watching people breathe, which sounds. But at any rate, so you could do a study on Alzheimer's disease or anything else with one of those monitors, get one answer, with a different monitor, get a totally different answer, because there had never been some decision made to determine what level of accuracy was necessary to actually be able to make those relationships count, right? I think what I'm hearing, actually, Andrew made it, I'm probably not as being as nice, I mean, not as saying it as well, but the fit for purpose of the monitor has to be, you have to kind of, it seems to me, you have to kind of know that, so that you know what your lower limit of, accuracy, of believability is. And I'm wondering if in any of these projects, and with respect to this even, have we considered what is the lower level, common denominator of accuracy that you would need to be able to even just do this for dose, not getting at Alzheimer's or even, diabetic complications, right? I mean, and how would you go about that, right? Because again, we keep talking about the device, but really what we're talking about is not the device, it's about interstitial glucose or blood glucose, right? And that variability. So what do we know about that, and how can we get that common denominator, that lowest level of goodness, accuracy?
Yeah, so that goes back to what's your ground truth and what's your reference, and long term, that is very, very hard to accomplish.
Well, Joe, maybe I can weigh in here. And the way I think about your question is it's one where a definition can really help us. When we think about a biomarker, we think about the biological feature, the rate of respiration. We don't think about how it's measured. And when we qualify it, we qualify the biological feature independent of the way it's measured. And so we can sort of separate the question out. So when we go back to CGM is not a biomarker. It is, the biomarker would be a feature, whether it is the integral over time, a high minus a low, whatever the particular feature turns out to be, CGM is how it's measured. Mm-hmm. And so the standards for measurement can be addressed sort of as Bakul spoke about, and as Jen spoke about this morning, where we need both analytical validity and clinical validity, but that sort of measurement question is independent from what is the biological feature of interest.
Joe, are you trying to make a comment? I think I heard your voice there.
No, I think this kind of gets back to the question of biological significance versus statistical significance and power calculations for addressing the signal that you're actually going to measure in a population.
Yeah, I think we missed part of that because we're not following the statement. Can you repeat?
I'm sorry. I guess the question really comes back to the context of use versus the statistical measures you're going to use to validate the endpoint. So if you're trying to understand the glucose monitoring data versus how a patient's responding or the number of subjects you're actually measuring, that's a statistical comparison for the feature you're measuring, not necessarily the clinical significance of the data that you're interpreting.
Right. Okay. So it's about the precision and accuracy of the measure specifically versus what that measurement means clinically for the outcome that ultimately you're seeking is what you're talking about. And yeah,
correct.
So
correct.
Separating the device performance from the clinical validation.
Correct.
Right. Right. Yeah, and I think the point that Andrea was making is that we cannot achieve a good clinical validation unless you have a lot of certainty in that in how that,
it depends
right?
what you need. You know, he actually brought up a good point earlier on the phone, is depends on what you're looking at too. Like if it's just to see like how somebody responds to food, maybe you don't need a certain, you know, the level of accuracy you might need if you're looking to, you know, develop, you know, making a decision on whether or not you're going to go to phase 2 or phase 3 of your drug. You know, which is, there's a lot of money involved in that, so you might want to look at the precision and accuracy with the higher level of scrutiny than you would for a different type of purpose. But it would be definitely up to you still to define. And I don't say any of these things to say that CGM can't be used in this way in the future, but I really just want people to understand that, you know, there is, you know, there's these devices that you're going to use to make decisions there are a lot of nuance to them. There's a lot of different variability that can be introduced, like we had mentioned earlier in the slides. Insertion site, you know, even, I mean, these devices even work differently in the hands of the user. These studies aren't that big. The studies that we're looking at, you know, the data that's obtained to support an approval or a clearance, I mean, they're like 150 to 200 people, but there are hundreds of thousands of people using these devices, and so is that really a good representation of what the actual device performance is? Maybe, maybe not. But you know, these are just things that you should really understand about your measuring technique when you're making decisions on that kind of stuff.
Okay.
Okay, it's late on a...
Just maybe one comment, because I think this is to me where consortiums make sense, or a non-competitive space collaborations make sense. And because actually these kind of validations would be needed anyway. I think we've been jumping into the space of digital measurements as a way to kind of prolongations of the clinical measurements being done in home settings. But this is a totally different story. I think there is a long established validity of what happens in clinics when this gets measured, and we're getting to the new space thinking that simply because we have tools, we can just do these measurements at home. I think we are lacking enough evidence of what those measurements tells us, which is different from what the measurements in clinics is telling us. That's probably that the space where a non-competitive collaborations could take place and have resources put together to help generate the understanding of what those measurements means, and then understand the limits and how can we use them for decision making.
Yeah, and one final comment with the consortium model, combining smaller number of patients, like we could collate them together and then capture more variability in that sense.
So it looks like a challenge has been issued to the Biomarkers Consortium.
And since this would be a metabolic disorders project,
I know.
Roberto, that means it goes right back to you. So this has been a very busy and intense, dense day. I am appreciative for everyone who's here and who's still here and is still contributing. And I think I'm going to take our time that I was going to use. It's on the list to sum up the day and say, I think we don't need to do that. I mean, we've had a lot of discussion. We've had a lot of really good conversations, which is exactly what this is supposed to do. And I think they may not be ready yet, maybe 15 minutes, but I'll give everybody a break before the reception, which will occur at about 5 o'clock or so, because that's when it's supposed to occur. So I appreciate everyone's day. Jen, do you want to say anything as a end of the day? But I look forward to seeing all of you bright and shiny tomorrow, bright and in the morning. So thank you very much for a great day, and thank you for everyone who's